My name is Lazzaro, and my project is measuring the efficiency of the metro system using the slime mold Physarum polycephalum. The purpose of my experiment is to test the efficiency of the metro system, and the current metro system, and its planned expansions using the slime mold Physarum polycephalum. About the slime mold, it is a fast-growing single-celled organism. When you introduce it to an environment, it will expand out and find food sources in order to survive. What it does is when it finds this food source, it will send out a network of protoplasmic veins. These veins carry enzymes, and when it finds a food source, it will use the enzymes to help it digest the food source. And it is benefit to the slime mold to make a very efficient network, because then it's going to be using the least amount of energy possible to carry the enzymes. Prior experiments, one of the first experiments that a group of researchers did is they used the slime mold to have it navigate a maze. What it is, they put it at one end of the maze and put a food source at the other, and it grew through and created one vein to the food source, and that was the shortest way through. They also analyzed the Tokyo railway system using the slime mold. These are the pictures of it. Procedure, the first thing I'm going to do is I'm going to culture my slime mold. I'm culturing it on 2% agar plates with oatmeal flakes as its nutrients. What I'm going to do is I need to subculture it once a week and create a backup, so in case one of mine dies, I'll have another culture to keep subculturing from. When I actually do my actual experiment, I'm going to put it in a Pyrex dish inside a box because it is photosensitive, and I will have a light shining up from the bottom of it and black felt on the bottom of the Pyrex dish that will only let light through in certain areas. This will represent a geographical constraint because the slime mold will not grow in that area because it is photosensitive. I need to decide where I need to place my oatmeals on the actual Pyrex dish, and that's I'm going to create a scale, and I'm going to use that using a map of the DC area. My camera is a low-lux camera due to the low light inside my box, so I've ordered that. Analyzing the data, the main thing I'm going to use is the computer program GIS. First measure I'm going to be using is the total distance that the veins take up, essentially, or they will represent the tunnels. I also seen in other experiments that scientists have used what they call fault tolerance, which is essentially if one of these veins is cut off, how many would a node essentially then also be isolated, and they compare that and the total distance to measure efficiency. Hopefully there will also be other use on GIS that I can use, and then I need to do more testing. Progress, I've ordered and received the camera, and I had to order the lens separately because it didn't come with the camera. I've also ordered and received my slime mold, and I've started culturing that. I've mixed my 2% agar and poured it onto 13 different culture plates, so I've now lined myself up for a couple weeks of culturing. I've also started to culture the slime mold on those plates.